Hello everyone. Today I would like to discuss an important topic in B.Pharm curriculum, that is antihistaminic agents. Before going into the topic, we need to know a brief introduction about autocoids. The word autocoid comes from the Greek words autos, means self, and akos means healing substance or a drug. These autocoids are substances produced by a wide variety of cells that have intense biological activity locally at the site of their synthesis and release. These autocoids are also called local hormones because these hormones act locally on neighboring cells or on the same cell that secrete without first entering the bloodstream. And these local hormones differ from other hormones like endocrine hormones, because endocrine hormones are circulating hormones. That is, they pass from the secretory cells into interstitial fluid and then into the blood. These autocoids are involved in physiological and pathological processes. So there are two types of autocoids. Amine autocoids, example, histamine and serotonin, and the second type is lipid derived, example, prostaglandins, leukotrienes and platelet activating factor. So this histamine was first discovered by Sir Henry Dale and recognized in the 1920s as a major pathogenic mediator of allergic disorders. And the histamine is a biogenic amine which is present in many animal and plant tissues. If we see the synthesis of histamine, histamine is derived from the decarboxylation of amino acid histidine in the presence of the enzyme L-histidine decarboxylase. So this is a diagrammatic representation how histamine will be released. So the first step is when an allergen comes in contact with a B cell, it results in the production of large amounts of IgE antibodies. These IgE molecules attach themselves to mast cells. The IgE primed mast cells release granules and powerful chemical mediators such as histamine, leukotrienes into the environment. These chemical mediators cause the characteristic symptoms of allergy. So coming to the storage of histamine, histamine is stored in mast cells in complex with heparin, which is an anticoagulant, and this histamine is also stored in basophils in complex with chondroitin. So these are the releasing agents of histamine. That is, these agents help in the release of histamine. And there are two types of releasing agents. One is IgE mediated releasers and the second one is non-immune releasers. So in the IgE mediated releasers, there are so many releasing agents like food, examples eggs, peanuts, milk, drugs, examples penicillins, sulfonamides, etc., venoms, foreign proteins and enzymes. So these are the IgE mediated releasers of histamine, and these are non-immune releasers. So under this category comes morphine and other opioids, aspirin and other non-steroidal anti-inflammatory drugs, vancomycin, succinylcholine, etc. Coming to the histamine receptors, these histamine receptors belong to the family of G protein coupled receptors, and there are four subtypes of histamine receptors: H1, H2, H3, H4. The H1 receptors are present in brain, GIT, CVS and lymphocytes. H2 receptors are present in myocardial cells and parietal cells. H3 receptors are present in CNS, myenteric plexus and gastric mucosa, and H4 receptors are present in spleen, thymus, T cells and eosinophils. So what are histamine antagonists? Drugs that block the action of histamine at H1 or H2 or H3 or H4 receptors are called histamine antagonists. And these histamine antagonists are of three types: drugs that inhibits the histamine release. Second type is drugs that inhibits the action of released histamine. So under this category there are three types: H1 antagonists, examples first, second and third generations, H2 antagonists and H3 antagonists. And the third type is drugs having dual action. So in today's class I have given a brief introduction about what are autocoids, what is histamine, how histamine is stored and released, what are the type of histamine receptors, and what are the examples for histamine antagonists. In the next class I will be discussing some SAR points, meaning structure activity relationship points. Thank you.